Hello everyone, my name is Sam Smith. I am a sixth former senior at Millbrook School and a member of the independent science research class. This year I studied the effect of COVID-19 prevention policies on the bacterial biodiversity of a boarding school campus. So as many of you, or as all of you hopefully know, this past year COVID-19 has been a pandemic that has swept the world, the nation, and Millbrook School. There have been millions of cases and hundreds of thousands of deaths as a result of this respiratory virus that is highly contagious. And in response to the pandemic, schools around the country have implemented various COVID-19 policies that are aimed at preventing and stopping the spread of COVID-19 around campus. These policies range from anywhere from mask wearing, glove dispensers in the dining hall, social distancing, and mandatory sanitizing of surfaces, among other things. And these, like I said, these practices are meant to slow the spread of bacteria on campus. So from these policies, I was wondering, how well are they doing at actually stopping the spread of the virus on campus? And because viruses are spread in similar ways to bacteria, I thought that judging the bacteria spread on campus might also tell me how the virus was being spread as well as a result of these policies. So I posed the question, how do COVID-19 sanitary policies affect the bacterial biodiversity on a boarding school campus? And I answered this question with the hypothesis, if COVID-19 policies are implemented, then bacterial biodiversity will decrease. To test this hypothesis, I collected bacteria at 4 stages throughout the year. These stages were at the end of the fall, twice in the middle of the winter, and once at the beginning of the spring, and at 4 different locations. And those locations are shown in Figure 1. They were the mask door, Math and Science Center door, the door of the Student Center, the barn, the front door of the dining hall, and the glove dispensers in the dining hall that were implemented this year. I chose to collect bacteria at 4 different stages of COVID-19 policies because the policies were different at each stage. This allowed me to compare the bacterial biodiversity at each stage and see which policies were effective and which ones were not. So once I collected the bacteria at these 4 stages, I did 2 things to them. I grouped them into different categories. There were 33 different categories based on their morphology, which means their texture, their shape, their size, and their color, among other things. And once I had those groups, I plugged those into a Shannon Biodiversity Index equation, which is represented by that equation below. And that equation basically gave me an index for bacterial biodiversity on campus. And I did that for each of the 4 stages, as well as for Millbrook as a whole in the 2020 and 2021 school year. Once I had those data, I compared it to Alex Chalk, a former ISR student's data from 2015 and 2016 school year. And that was used as a benchmark to see how COVID-19, or how the bacterial biodiversity changed in a year without COVID-19 policies from when there are this year. So in all, 42 different colonies were discovered across 4 stages. 19 of those were found in stage 1. 7 of those were found in stage 2. 12 of those were found in stage 3, and 4 were found in stage 4. In terms of the Shannon Biodiversity Index, stage 1 had the highest with a value of 2.638. And stage 4 had the lowest with a value of 0.563. Stage 2 had a value of 1.154, and stage 3 had a value of 2.095. And as for Millbrook School as a whole this year, our bacterial biodiversity index was 3.098 compared to 2.29 in 2015 and 2016. So the Shannon Biodiversity, that's also represented in Figure 2 here. So the Shannon Biodiversity Indexes for each of the stages imply that there were some stages with more effective COVID-19 policies than others. Stage 2 and stage 4 had lower bacterial biodiversity than stages 1 and 3. Most of the COVID-19 policies at these stages were similar. There's one discrepancy that is clear between the policies at each of these stages, and that is the quarantines that occurred before the stage. In stages 2 and 4, there was a quarantine or a period where students could not freely move around campus before those stages. And these stages were the ones with lower bacterial biodiversity. This implies that because the other COVID policies were so similar across all stages, that the quarantines had the largest impact on the bacterial biodiversity around campus. In terms of the entire school year, our Shannon Biodiversity Index this year was higher than it was in 2015 and 2016, which implies that although some of our policies were effective this year at stopping the spread of bacteria around campus, as a whole, they were ineffective. Bacterial biodiversity is the relative abundance and species evenness, so the species richness at a location. And for the bacterial biodiversity to be higher, this means more bacteria as a whole were allowed on campus, as well as more species. This means that because our Shannon Biodiversity Index was higher this year, we allowed more bacteria and more types of bacteria on campus with our COVID-19 policies than in a year without them. This means that they could have been ineffective in stopping the spread of bacteria and thus stopping the spread of COVID-19 on campus this year. Thank you.